Well, since we are about to complete the part of plant tissue culture, I already mentioned in my last class, the last to last class is about the topics that we are going to review, right? We are going to talk about, and I'm not sure, but possibly I'm going to change the date of the exam for the second partial because I would like to insert some few things in the discussion. And let's see if I can make some changes. I will let you know, but possibly I will let you know if the exam date changes. It's not certain, but I will let you know if I'm going to change. Okay, it depends on that time management. Okay. Anyways, so the last class we had seen a very important technique, right? Protoplast culture or somatic hybridization. Do you have questions and doubts about it? Or do you want to comment anything? You want to clarify anything? Yes, Carla.
Yes, you do the somatic hybridization with the chloroplast, right? Just to be clear, we are doing that process with the protoplast. Yes.
Somatic hybridization?
Yes.
Yes.
Okay.
Of course. Just try to, sometimes, that helps us when scientific names, biological names, they says a lot. Somatic hybridization. Somatic means somatic cells, right? Which are not germs and normal cells. Anywhere from the body except the fertile cells or the reproductive cells, okay? Except sperm and ova. So everything is somatic and hybridization. How we are going to make a hybrid by the fusion of protoplast, okay? So somatic hybridization is to develop the varieties, right? Develop of the variety by mixing two different plant cells, which may be different, right? Different species, even different genus, right? Which is not naturally possible, right? Because there is no genetic compatibility. Naturally, they are not compatible. They do not fertilize, but you can fix this issue by using protoplast, okay? Doubts, questions? Any other comment? Yes, Emilia, then Juan Luis, then Barbara.
Yes, teacher, and I was checking the notes that the presentation, and in some point in a graph you put that the result of the mitosis are hybrids and synkaryons, I think. And in another slide you put that the result of the fusion is hybrids and hybrids. Hybrids and synkaryons are the same thing?
Yes, they are same, of course. There are only two kinds of, fusions, possible fusions. Well, there are many, but mainly two. The fusion between the cytoplasm, so that is called hybrids, right? When the cytoplasm is playing an important role because hybrids or cytoplasm become very relevant because in plants, they have two different genomes that are functional genomes, and they have genes that can be that can have important effect, right, in the plant. So that is why cytoplasm is very relevant. If it is going to be any other species, maybe human, but cytoplasm does not have much impact because most of the mitochondrial genome is silent. It's a non-coding reason, right? But in plants, the plastidical chloroplast are very, very active genomes, okay? So hybrids means enucleated. Enucleated means a cell which does not have nucleus mix with a cell with the nucleus, right? So that becomes hybrids. And the differences between which cytoplasm or which one of the three genome is going to mix, that could be little variable. In question of the hybrids, when you call hybrids means the main genome of the plant or any other organism, it is in the nucleus. So that is why the majority of the hybridization or the most efficient recombination will take place into the nucleus, right? Nucleus is the machinery of genetic composition. So basically what will happen that maybe 95% of your characteristics that you're looking for, they are in the nucleus, and maybe the 5%, there is no approximate, but I'm just trying to, figure it out, like 2 to 5%. So 95% of the genes, they are in the nucleus, right? So the hybrids Then hybrids, it could be from one cell to another cell, but the nucleus is going to be fused. Okay, so these are the difference. And yes, the hybrid, whenever makes the terminology the same, Emilia, for those, both of them.
Okay, thank you.
You're welcome. Juan Luis?
Yes. Well, my question is about when you make the hybridization and you make like hybrids instead of hybrids, then you have a multinuclear cell, or there is also a fusion of the nucleus?
Of course, very good question. You can have both of them, Juan Luis. Okay, both of them are possible. But when you have two nucleus separated, most of the time the cell will not survive, or will have a very unexpected, or how to say, the irregular growth. Okay, because if they are not fused and every nucleus is going to work independently, the cell become crazy. Okay, in a very simple term. Okay, so your objective, if you have those nucleus separately functioning, that is generally not possible, because only one nucleus is supposed to function. Okay, because the cell will divide. You understand that? And to divide a cell, the nucleus has to be one. If there are two nucleus, the cell is not going to be a cell that can be divided. Okay, so the fusion is the ultimate target. Okay, if there is no fusion, you are not going to expect the variations and the characters you're looking for. And everything you are doing is just to mixing the genes, the alleles in one genome. Okay, in one genome. That is the idea. If you have two different the separated nucleates out there, if they are not fused, it's very difficult. The cell is going to divide. Okay. Generally, those cells you are going to eliminate. Okay, most of different kinds of fused product you are having, you will only isolate or choose to cultivate which are already hybrids or hybrids, right? The rest of them you are going to eliminate, because they are going to take your time, reagents, and every effort, but they are not going to give you any product. Okay. And for the selection, there are so many different things, and that depending on the protocol, depending on the particular objective, the selection can take place by the antibiotic, by the herbicide, by the pesticide. And protoplast thing is very interesting, but it's always a risky method, because there is no certainty, right? There's no certainty. The certainty is when you are very, very sure and you have the very precise information of both of the varieties or species. Okay, so then you can find out the right screening method. You can find out that what kind of hybrid or hybrid is going to work for you. If you know that my gene I'm looking for, it is in the mitochondrial genome, not in the nucleus, then you go with not the hybrids, you will go with the hybrid, isn't it so? Because you are looking for your gene to be expressed by the mitochondria. Okay. Did I answer? Okay. Any other doubt question? Because I'm going to start the exam on it. Okay. So ask before the nuclear bomb comes to your head. No, nuclear bomb. It's a chocolate bomb. Okay, don't worry. Yes, Emilia.
Teacher, what was PEG?
PEG is a polyethylene glycol.
Okay.
Okay. It's a chemical that make the cell membrane porous. Okay. By binding to the cell membrane, it do the ionic interexchange between the cell membrane, and it make it porous. Okay. Everything to become fused, all the fusogens, they are basically making the cell membrane porous. Calcium ions, PEG, sodium nitrate, or electroporation, right? Physical methods, all of them just to make porous your cell membrane.
Okay, and in this case, well, we really use this type of method, the induced fusion and the electrofusion, right?
Electrofusion.
Fusion and electrofusion, right?
Exactly. Okay. Chemofusogens and electrofusions, right? Electrical fusion. Any other question, doubt? Okay, so I'm going to start with a Kahoot, and after the Kahoot, we will start a topic which is called suspension culture and Harry-Wood. But before going there, did you read the article I shared with you? Sure? Okay.
Yes.
Okay, so there must be so many doubts and questions about it, right? So we will discuss about the suspension culture and Harry-Wood culture. That is going to be the topic for today's class, okay? And after the class, I will put one exam on the same topic in Socrative, okay? So I need your attention. And that is a graded exam, okay? And finally, we will see. And well, there will be no homework for this weekend, okay? Just to make you little relaxing and happy, so that on Monday you can send me emoticons that are a little bit more exciting, okay? Okay, cool. So let me start the Kahoot, and let's begin with the quiz. It's a very small quiz, but yes, it's important to recall all the fundamentals about the protoplast. Okay, coming to you. Okay. Okay. Music. What music you want? I'm your DJ. Academic DJ. Okay. So this is only 20 questions, okay? And oh, my internet, hopefully. Okay. Okay, let me. Okay, this is the PIN 995-3130-3103. Okay, 1 second. Let me share the music, and you all are in, I guess. Here I go. Thank God. This is finished. I was about to sleep with this music. Bad choice, meditational music. I was about to sleep. Oh my gosh. Okay. Anyways, I could see a lot of doubts, isn't it? A lot of doubts. I didn't see not a single question that was answered perfectly, right? There's so many doubts. So the quiz is uploaded on the Canvas. So please have a look on it. And if you have a doubt or question, we can discuss and I can help you in clarifying your doubts. Okay. Oh, for the next time I have to put some music that is little exciting, right? Okay. So now coming to the topic. Okay. Okay. Let me see if you really read about it, right? Okay. So the topic is, you know already, if you say so, that you have read about it. So this is the suspension and Harry-Root culture system, okay? And the main application of both of them is the production of secondary metabolites by biotechnological means. So tell me, what have you read? What you understand? What you see? Any comment? Anything you know about it? Yes, Carla.
That even though it's not like popular for normal tissue culture, it is a really good alternative when you are planning to do subcultures or you are really looking for a little bit more of like manipulation during the tissue, the micropropagation, or in this case, the second metabolite production, because with suspension culture, you have the advantage that you don't disturb or damage a lot the explant or the material you are working with. And also it can lead you to a better mineral treatment than with normal medium. So it's a really good alternative, but you have to really be conscious of what you are trying and looking to investigate and obtain, right?
Exactly. The application, obviously, you mentioned that maybe this is, well, anyways, this application is very, very important. And you can say the only application that allow you to exploit the plant cell biotechnologically, okay? A variety of medicinal compounds, medicines have been developed by this. The first class I mentioned, about the compound chikungunya that was patented by Pfizer in 1952. And that compound was produced by by the suspension culture, okay, by bioreactor based on suspension culture. Okay, so this is the most important biotechnological application. Okay, what else? What else, guys? Okay, any comments?
Yes, teacher, I have a question. For example, in the
questions. Question I will take when I will start the topic. Can we wait some few minutes for the question? Is it okay?
It's a question about the reading.
Okay.
Do I also wait or can I ask?
Okay, go on.
Okay. In the reading, there says that the twin 20 helps for the alkaloid movement from cell to the medium, but I don't know, I don't understand why that would be useful.
What thing?
It says that twin 20 is
twin 20,
used to...
it's a detergent. It's a detergent.
Yeah, but it says that it's used to move alkaloids from the cell to the medium, and I don't know why alkaloids are important here.
Alkaloids is a kind of secondary metabolites, and the T20, what it does, it's a phospholipid, it's a detergent which allow the cells to become more permeable. When they become more permeable, they release the alkaloids outside the cells. So that is why it increases the extraction efficiency of the method.
Okay, so here we would be interested in retrieving these alkaloids.
Yes, releasing the alkaloids, because production is one thing. Then you have to think about the extraction and purification, and that is not basically part of tissue culture. That is the more about analytical and purification methods, right? That I will also explain. I will also explain the class. Okay.
Okay, thank you. Yeah, I just have that question. Thanks.
Okay, no problem. You're welcome. What else? Any other comment about the technique? No? Okay. So what you are seeing here, okay, Johnny, go on.
Yes, teacher. I read that the suspension also, as Carla said, works for the production of many metabolites, but the secondary route, the Henry route, is used because there are chemicals that are hydrophobic, and these chemicals can go out of the cell. So you induce the Henry routes in order to increase the yield and other stuff.
Exactly. And hairy roots you have to induce because when your compound is produced in roots, isn't it so? If it is produced in the leaf, then you will do suspension culture of the callus and taking out, because they are two different things. Suspension culture and hairy root culture, they are two different things, okay? And don't mix it, because hairy root is producing roots only, okay? The production of root in the liquid media and the suspension media, that is called hairy root culture. And suspension culture could be anything. Even the hairy root culture can be considered as a suspension culture, but that is not the case normally. Suspension culture is mostly when you cultivate cells, okay, into a liquid media. That is called suspension culture, not hairy root culture. Okay, so that is the thing that you have to understand. Okay, in this picture, you are seeing a lot of roots. Okay, a lot of roots, a bunch of roots, and you can see they are growing like in a very natural way. Okay, and there is no stem. That is the most interesting thing, and how this is possible molecularly and biologically, how this is possible. Here you are seeing a media. In this case, you are seeing a media. This is basically MS media, right? Morschwag-Skoog media, but without agar. There is no agar. So here you are seeing basically a lot of roots growing day and night. Okay, so the volume, you are interested in increasing the volume. Anything, because you know secondary metabolites are produced differently. Okay, differently means they are produced by different parts of the plant. They are produced in leaf. They are basically produced in every part of the plant, but they are distributed according to the necessity of the plant. Where the plant needed them, plants produce it. Okay, so my secondary metabolites, which I'm looking for, if they are being produced in the roots, then definitely my objective for the production, commercial production, is going to be focused on the root culture, because my objective is to create a lot of roots. So I have the enough amount of biomolecules and I can produce and manipulate and control these conditions. And that is why I use bioreactors. Okay, so that is one of the most important difference. Okay, so here you are seeing in the suspension culture, a suspension culture is nothing but in a normal tissue culture media. This is the only thing you are adding and removing here is the agar. Here you are seeing agar, and this is the normal flask, and now you just do not add agar, and now this media become liquid. And you see there are some kind of green material, and this green material is telling that there are cells, okay, in the media. So that is called suspension media, the right suspension media. When you will find roots over there, you will tell it hairy root culture. Okay, but both of the things are going to be happen in the suspension culture. So the basic idea about doing suspension culture is to exploit it by controlled method, and that is the bioreactor system. Okay, that is the main idea. Okay, and the second idea, that you are growing cells or roots. So basically they can grow into the liquid media, but the liquid media, if you do not agitate, if the liquid media does not have gases, which is CO2 and oxygen, the cells will die. Okay, they will suffocate, because it's a plant cell. It's constantly need nutrients. It constantly need the right amounts of the nutrients and pH and the gases. So that is why the liquid media helps you to adapt this system to the bioreactor based system. Okay, so that is why this is very important. And if you grow callus, you know, in solid media, if you put in the liquid media, the callus is going to be suspended, right? It's going to be make a pellet down there on the flask. So that is why agitation is constantly required, because you do not aggregate these cells. Okay, so you have more homogenized media and more homogenized cells. And you don't want your cells to coagulate. You don't want your cell to become aggregate, right? Because you need every cell has to be separated and growing well. Okay, they can multiply, yes. They can divide, yes, but you don't want them to transform. You don't want them to differentiate, because if it is going to be a normal callus, what will happen? If you put the hormones, they will transform in something else. It could be an embryo, it could be an organ, whatever it is, but it will start the differentiation process. That differentiation process is blocked when these cells grows in the liquid conditions. Okay, and you are not adding any phytoregulators. That is also very important point. Okay, because if you add phytoregulators, what will happen? That your cells will start transforming and start differentiating. Okay, so how to achieve an ideal suspension culture? Okay, the ideal suspension culture basically can be achieved depending on the obviously the methods you are adapting. Everything starts from a callus. Okay, callus is the main source. Okay, if you have callus, then only you can have the suspension culture. Then you have to adapt the media, right? You can check the same media you are using here in the solid conditions. You can use it without agar. And this callus you are going to disaggregate, okay, by physical means, and by centrifuge, or by just by breaking it by the shocks in the shaker or by the agitations. So they start disaggregating, and the bigger clamp or aggregates you will throw out, you can even filter them, and only the more homogenized things you are going to add it. Now these cells will grow and multiply. Okay, and finally these cells basically could be used for other purposes. Okay, not only one thing, they can be used for many other purposes. Okay, so then you can go with the embryogenesis, you can go with other kinds of methods. Okay, the normal protocol, you know, this is the whole protocol. I'm not going to tell you step by step things, you can read it, but the whole protocol is obviously it's focus number one, the established tissue, the callus tissue. First, you have to establish. You can take one piece or any amount of the piece. That is not any standard about it, but general, this protocol is based on the 1 gram of callus. Okay, then you can go with the this callus. You can cover. You can put in the anything that make it humid. Okay, in the cotton, in any place where you can put, so it is not going to dry up, because if you take the callus out from the media, it becomes stress. Immediately it will become a stress, because it is already stressed. Callus is a stress condition for the plants, but it will become even more stress. So you can just not take the callus and put it 5 minutes in the open air or 10 minutes in the open air. You are going to kill basically most of the cells, okay, within few minutes. So it has to be very, very obviously aseptic, and it has to be very weight. Okay, the tissue paper or the place you are putting, it has to be weight. Then you can break these clamps. Okay, manually, this is also possible. By any mechanical support, by scissor, by any metal material which is sterilized, you can break it, or you can use heavy shakers, okay, which moves on this speed. Okay, so this is like 80 to 120 RPM round per minute, and the shaker what will do that it will disaggregate cells. Okay, not immediately. If you are doing immediately, you will kill many cells. So it is a process. It will take like 1 day, 2 days, in some cases up to 7 days to disaggregate whole callus and make your media more homogenized. Okay. Well, this is after that the filtrate, the final filtrate that you have will after few days, okay, depending on the method, will have these free cells. Okay, and cell aggregates. So if you have a lot of cell aggregates, you can filter, okay, on a metal sieve, Okay, everything has to be sterilized, okay, 100%, and this all done in the laminar flow. So you can just put the media, you filter on the coladora, okay, or sieve, and you can make it more homogenized, okay. After that, you can again filter it, and this filtrate which you have, you can centrifuge for depending on the protocol, depending on the types of cell, but from 10 to 15 minutes, and you can centrifuge by 500 to 1000 RPM. Even lesser, in some cases only 200, because if you do if you use centrifugation on a very high rate, they will become very compact, and again you are going to have a problem. They will be aggregate very forcefully, and obviously the viability will also get affected. Okay, and finally you can pour the supernatant and have these cells. Okay, now you got your cells to put in the fresh media. Then you can go and resuspend this pallet which you already have, right, in a fresh liquid media. Okay, no agar. And then you can make 1 or 2 more flasks, because you cannot produce sufficient amounts of medicines or any biomolecules in 1 flask, 2 flasks, maybe for a small research. Okay, if you want to produce your biomolecules in grams, in kilos, then the bioreactor size is going to be very big. We are talking about 10 liters, 50 liters, 100 liters, 1,000 liters, 50,000 liters, 100,000 liters bioreactors, right? Depending on the necessity of the biomolecules. Okay, it is for the research or commercial production. So basically, the flask is, you are going to multiply every time. The cells is going to keep dividing in the media, and you can keep, just maintain a very big amount of the volume. Okay, that is very important. And at the same time, you are going to keep monitoring that your cells are alive. You can use the cell cytometer, which you can count the cells. You can also check live and dead cells within it. So a lot of parameters has to be considered. Okay. And at the next subculture is when you are taking the same thing in a fresh media. Okay, any explant, any material in the fresh media, that is called subculture. So you repeat the previous step by many times. You can keep repeating whenever you have to put in a new media, it becomes subculture. You can have 10 subculture, 20, 40, 70, depending on the protocol. The idea is to have the right amount of liquid for the final process. Okay, you can produce the biomolecules in this stage, because cells are going to be produced. Cells are producing the metabolites constantly, and it could be intracellular, okay, intracellular, and it could be extracellular, okay, extracellular. Intracellular, when the compound is being produced inside the cell, and extracellular, when the compound is being produced and released in the media. So that is also very, very important and relevant to establish your method. Well, anyways, after several subcultures, depending on the requirements, transfer the 10 ml of cell suspension to each flask, okay? And then you can eat like 300 or 3 milliliter fresh media. So you have many other flasks, okay? So what they are doing here, they are increasing the volume, okay? And finally, to prepare a growth curve of cells in suspension, transfer and defined a definite number of cells measured accurately by hemocytometer. Okay, hemocytometer, I will tell you how to count. We will learn about the hemocytometer, how to use it very well in the tissue culture, animal tissue culture part. Okay, so don't worry about the hemocytometer right now. But yes, it's a very small the glass plate which help you to counting cells manually. And finally, the pipettes out every the aliquots, right, in the short intervals of time, 1 to 2 days, and count the cell number. So this is just to keep monitoring that your cells are duplicating. Your cells, you are maintaining cells alive, because in hemocytometers, you can clearly differentiate dead cells and live cells. So if you're taking, for example, 100 microliter of drop, this 100 microliter of drop will tell you after 2 days that your cells, if you are doubling, not doubling, the number is increasing, not increasing. So that way you can have the control, and you are always prepared, to improve the conditions. Okay. And after that, plot the cell count data on the passage on the graph paper and curve, and finally you can got your growth pattern. Okay. So this is a simple thing I will let you know in animal cell culture, because in plant tissue culture, basically the growth curve is generally not required. it is only required when you are doing this thing, okay, when you are doing the suspension culture. In any other method, it is not required, but animal cell culture, every time you are doing, all the time you need to make growth curves, okay? So we will learn about it. So here, just to understand by the graphic, this is the tissue, okay? This is the callus. You know how to create callus. Then this callus, you put in the liquid media, right? This is now basically the liquid media. These are the shakers. So this clot, this clot is going to be homogenized. And after this few days or few weeks, depending on the method, it become more homogenized, okay? Then you can filter this, okay, to remove the bigger aggregates and to have more homogenized media. So this is your pet. This is any flask, anything which you have the more homogenized things. Now you will centrifuge, you will discard all this supernatant, and this is the pellet that you are going to resuspend in the fresh media. And now you will take the small amount. In hemocytometer, generally you take 100 microliter, okay? 100 microliter, and you can count the cells, how many cells are alive, so by which you can keep monitoring. And once you check the cell numbers, now you can again put them in fresh medias, in a small aliquots, and try to double and triple and quadruple the amount of the media. Finally, you can plate these cells, okay, in a solid media, okay, yes, solid media, because now this become embryogenesis. So basically, this is the method by which you can do embryogenesis, or you can start from here the bioreactor processes, okay? So after that, now you will put the picogram. On Monday, I will give you the protocols about embryogenesis. So this is about embryogenesis. You have the picogram, the hormones that is very famous for the inducing the embryogenesis. Picogram, it's an auxin, okay? It's an auxin, higher concentration of auxins, and you have to keep trying. So once you got the embroid, you are lucky, you got the embryogenesis and you have your protocol, okay? If you don't want to do this directly from here, these cultures can be used for the production of secondary metabolites in the bioreactor systems, okay? So that is the idea about this technique, okay? Yes, questions, doubts?
Just one.
Yes.
What is the thing on the top of the flask? The thing that looks like a strawberry.
It's a algodon. It's a cotton. It's a cotton. This is called cotton. Is the name?
Like a toronda?
No. It's a cotton. It's a. I forget the name.
It's a cotton plug, right?
It's a cotton plug. Cotton plug. Yes, thank you. It's a cotton plug. We call it cotton plug. It's a made up cotton, compact cotton. Okay.
That's for gas interchange, like so it can breathe in a way? Yes.
You can use cotton plug. That's a very traditional thing. Now even you can use plastics, but cotton plug is, maybe we will try to have some demonstration, but you just compact the cotton as compact as possible and just use it. So yes, the idea of using cotton plug, that it helps you to maintain the gas exchange and avoid the bacteria. Okay. Okay, got it?
Yes, thank you.
Okay, so now try to understand this, and this is very, very important, okay? So this is the process. For example, in tobacco produce so many different secondary metabolites, right? What is the secondary metabolites that is very famous in tobacco?
Nicotine?
Yes. So nicotine, for example, or tobacco is also a very famous anti-insecticide, okay? It can be used, there are so many compounds that it produces as insecticides. So for example, this is the first step. The first step may be the anything, but it is about the callus induction, okay? This is the part you are seeing here. This whole process is about to doing transgenic or transformation by genetic modification, right? So that is something which is, because tissue culture is a transversal technique, and the development of the transgenic use this pathway. But we are not talking about the development of transgenic here, okay, just to clarify. So you have the callus here, right? This is the callus. Finally, now this callus is green because you have very nice homogeneity, right? And finally, you can transform or untransform. That is not necessary. You can transform by bacteria, by other kinds of matter to make it transgenic, but that is not the idea. But yes, finally, when you have this, look the amount and the size of the flask. I told you, 100. So here, not less than 100 flasks are there, right? Because you are going to produce a large amount of nicotine. Nicotine, basically, it's in most of the cell, it's extracellular, okay? So what it means that your nicotine is already in the media, okay? You don't have to rupture the cell. You don't have to isolate the cell. So you can make it more continuous, right? What you have to do, you just take out the media, right? And put the fresh media. Take out the media, put the fresh media, and you can make a continuous batch, okay? And this media can be, this is the bioreactor, automatic bioreactor, and the isolated media can be further purified by HPLC techniques, okay? And that is called downstream processing, okay? Downstream processing means the purification, okay? The polishing of the product. And upstream processing, everything you are doing from here, okay, from here to here, this is the upstream processing, means the production process. So downstream processing, obviously it is HPLC by many different kinds of SPLC or chromatography method, affinity chromatography. There are so many different kinds of chromatography method. We are not going to go into details, but the most important thing that the extracellular, if it is extracellular, you are going to extract the media, and the media is going to be purified. What will happen if it is intracellular? Then it becomes a little complex because you have to take out the cell. That means that you have to stop the process, right? Basically, you have to stop the process. You cannot keep producing continuously, or you have to divide at least half the process and take out the cells, make a centrifuge, release and throw out all the suspended media, take out all the cells, do a sonication. After the sonication, put some other media, and after sonication, you rupture the cell and release the secondary metabolites. Then you again centrifuge so that your secondary metabolite is going to be in the suspension. Now this suspension or suspended supernatant then can be used to purification process for the downstream processing, okay? So it's very important to keep in mind that everything depends on the intercellular and extracellular molecules, okay? This is the another case of this plant. I forget the local names, but Kurwa. It's another medicinal plant. Here you can see what you are doing is basically organ culture. After the organ culture, you are developing the callus. After the callus, you are going to develop the flask and make suspension culture. And now you are going to grow the roots, okay? Now you are going to grow the roots, and now things are changing because here, now you are converting the suspension culture in the hairy root culture. And hairy root culture is a special phenomena that is supported and done by a bacteria that is called Agrobacterium rhizogenes. Agrobacterium rhizogenes has the capacity to accelerate monster growth in the roots, okay? There is another brother of the Agrobacterium rhizogenus that is very famous for the transgenic processes that is called Agrobacterium tumefaciens. They are very similar, but they do very different function. Agrobacterium tumefaciens is used to transfer the foreign DNA or for the transformation processes in any part of the plant, okay? But Agrobacterium rhizogenes has a very special function. It will only promote the growth of the roots. So what you do, you just inject some of the bacteria inside the root, and this root become monstrous. It will keep growing and growing and growing and growing, okay? It'll never stop. So once you have the enough amount of the root, you can see a lot of roots here, right? In a bioreactor. So there is a lot of root. They are supported by any material. And now you can produce same thing. intracellular or extracellular gain. Okay. And then in this case, this is the compound there. This is a famous medicine. It's a picroside 2, then they purified picroside 1. Okay. So these are the final compound that is going to produce, and it's a medicine. Okay. So now you understand the difference between the suspension culture and the hairy root culture. Okay. This is the chiconin. I mentioned you in the first class. This was the very famous compound. It's for neuro disorders. First biotechnologically produced compound from plant. Okay, patented in 1952 by Pfizer. You know, Pfizer has more than 1,000 biotechnological patents. So this is a bioreactor, and this is the cells that produce chiconins. Now it is red because the plant cell is red. Okay, so don't get panic. It's not blood. It is not agua de Jamaica. It's basically the cells that is red. Okay, it's a very less chlorophyll. It's carotenoids. Okay, you remember carotenoids? Another the different kinds of chloroplast molecules, chlorophyll molecules, chlorophyll B. So this is how they keep producing. This is they are producing now, and they will extract it, the whole media, to purify the chiconin compound. Chiconin is very famous. It is in the market. Still, it is many companies sell it, and very well commercialized. Okay. So the importance of the suspension culture. Number one, basically you can understand, you know, all the basics, biological functions about the metabolic pathways, the cell physiology, its metabolic rate, metabolic activities, biochemistry, even structures and all that, right? That can be done. The best application that they can be used to produce secondary metabolites. Okay, the only way by which you can produce large amounts of the medicines. And also the mutagenesis can be done in some methods. You can very easily control, you know, here you can put even UV light, for example, right? UV light to induce some kind of mutations, or you can put a red light on blue light. That is also a new techniques nowadays. You can put light bulbs over here, blue light and red light, and they will also induce and increase the productions or any different kinds of property that is not possible naturally, right? So there are so many different kinds of alternatives that can be used to promote the productions. And obviously that it make the whole process very, very simple and completely under control. Okay. Now coming to the hairy root culture. Okay. So hairy root culture, basically, this is a very nice picture. Okay. And this is from a plant, and you can see these roots, they are extremely hairy, right? A lot of pellusas. Okay. They are extremely heavily hairy, right? They are hairy because you have infected them with the Agrobacterium rhizogenus, and they will not stop. You cut the plant, you take out the plant, so they will not need plant part. Any aerial part is not required. They will keep doubling. They will keep increasing. They will never stop. Okay. So that is the idea. That is the idea. And our best friend here is the Agrobacterium tumefaciens, Agrobacterium rhizogenus. This is the bacteria. This is the natural genetic engineer, natural biotechnologist. Okay. And it has a special plasmid for doing this thing. This is called tDNA, very similarly to the Agrobacterium tumefaciens. It is capable to insert the genome directly into the plant genome. Okay. Naturally, you don't have to do any molecular biological technique. You just, if you want to do any transgenesis, you can put your bacteria inside the bacteria. Okay. You can put your genes inside the bacteria, and bacteria will take this gene as a vector and transform and integrate your genes into the genome without any problem, right? But in this case, we are not talking about transgenesis, but we are interested in production of secondary metabolite. So you just infect it, they convert the cells to uncontrollable growth. Basically, it's become like a cancer. It will keep growing. And finally, these cells can be used for the production processes, okay? This is the genetic composition, genetic structure of the tDNA, and this is the whole plasmid. This is called RI plasmid, okay? Rhizogenous plasmid or root-inducing plasmid, root-inducing, okay? And this is the tDNA have different kinds of the regulator sites like rol A, B, C, D, replication A, B, C, D. And these replicative sites helps in the gene expression of the tDNA. So it has the BIR region, which helps the bacteria to infect the roots, and then have the opine catabolism. The opine are the ones that helps the plant roots to grow massively. Okay, so this is the basically the whole machinery, genetic machinery that this bacteria have, by which it can initiate the induction process. Okay, so for example, this is the bacteria, this is the plant cell. In the bacteria, you have everything like any other bacteria, but it has this special thing which is called RI plasmid, root-inducing plasmid, okay, which BIR genes, virulence genes that infected, and tDNA, which integrates. So this is the T strand. This is the whole strand that going to be inserted automatically by the T complex channel that is available on the plant cell membranes. And finally, it is going to insert into the cytoplasm. And after that, it migrates to the nucleus and dissolve into the genome. Okay. So in a general process, you are going to bound the plant cell, means you are going to create any kind of injury, right? And then this is like a signaling. Okay. This is like a signaling. So bacteria can infect. Then you put the bacteria over the tissue. Okay. So the bacteria now will start working. The bacteria will going to transfer its RI plasmid, and then the integration will take place. And finally, after a few weeks, you will see that your roots will start creating, growing massively. Okay. The difference between TI plasmid and RI plasmid, because TI plasmid, I told you in my first or second class, the tumor inducing plasmid that induce calluses, right? But RI plasmid is irrelevant here. So basically they are brother. They are like hermanos or hermanas, however you can put the gender, I have no issue. So TI plasmid, for example, it is basically to induce the tumors, okay? Or a disease that is called crown gall tumor. RI plasmid, basically it's to infect the plant to produce hairy roots. Okay, this is the basic difference. This is the bacteria, Agrobacterium tumefaciens, Agrobacterium rhizogenus. There are so many different strains available. They are both available in the soil. They are soil bacteria. Okay, anywhere they are worldwide bacteria. They can find and isolated from any soil. At the size, you can see different sizes. This is smaller, this is bigger. This induces this thing. This is the hairy and less studied compared to RI plasmid, and this is very well studied. Okay, these are the basic differences. This is a very good picture. I like it much because it basically tells you that how the whole process of root development is regulated by different types of hormones. Okay, naturally, not directly in vitro conditions, but yes, naturally by the bacteria. Okay. So you can see the primary root, the lateral root, and the hairy roots. Okay. So whenever you are going to cut, you are going to have the primary root, you are going to have the lateral root, and then you will have the root hairs. Okay. So you can see how many different hormones are going to participate, right? It has many different hormones that are going to participate in major and minor role will be there. Okay. And many of these we do not know, but they are basically induced by the bacteria inside the plant. Okay, and even produced by the bacteria itself. So basically you can see the auxin biosynthesis. Obviously it's very, very, very important. Then auxin comes out. Then auxin induce the transportation and distribution of different kinds of these compounds, abscissicate, thylline, cytokinins, polyamines, strogalactones, gibberellins, jasmonic acid, brachytenoids, all of them are plant hormones. Okay, many of them you already know. So, but you understand that the auxin is the mother of every hormones, basically not mother, but basically it's the monitor and the main regulator of all kinds of combination that can happen in the plant system, right? And finally, the auxin signaling will take place and it will induce the root development. Okay. And here you can understand the jasmonic acid. Jasmonic acid is going to help you in the lateral root. It is going to in the primary root. So this is the article which was published by Saini et al. In 2013. And where they're just trying to elaborate that how many enzymes are involved, how many hormones are involved in the productions of the root, okay, by the rhizogenous bacteria. Okay, so you can have a look on it and try to just find out the relation between these the phytoregulators, okay, different kind of regulators, how they coordinate this whole process. It's a very complex process, okay? And simply, it has been drawn in this picture. This is the normal plant, okay? And then you cut it and then you will keep growing, okay? They are the infected, and you can see that how they just keep growing and keep growing. If you do not give them right space, right amount of gases, right amount of media, nutrients, they will start shrinking, they will stop dividing and growing, okay? So this is the plant, which is Gentiana scabra, okay? And this is the basically the whole process, okay? But here I'm including the transformation part, okay? Which is sometimes necessary, sometimes not necessary. The whole process, it could be anything, right? It could be bounded leaf, epicotyle, anything could be, right? And then you infect it with the Agrobacterium strain, right? This is the co-cultivation and direct inoculation. Basically, you can put into the media, into the media. So this is a co-cultivation, or directly you can cut the plant and put the bacteria on it. Okay, so that is also possible. Then obviously, depending on the antibiotics you want to use or not. And then the neoplastic roots at the infection sites. These are the first kind of roots that created. After the subcultures, suitable media, you have root cultures. And this is the transgenic part, okay? If you want to see whether it is transformed or not. For that, you have the tDNA, which has your own desired gene. And by PCR, by southern blot, you can identify whether your gene is inserted or not. Okay, that is the normal traditional technique for the plant transformation processes. Okay, and indirect detection, you can find out the chemicals. Okay, whether your chemicals are being produced. Okay, opines, because opines are produced by the bacteria. So if your plants and the media is producing opines, means that your cells or the media, your explant is infected. Okay, so this is just a validation part. Okay, now taking some questions, doubt, if you want to clarify it, anything. Yes, Carla.
Yes, like for example, from the same cells, can I get intracellular and extracellular components at the same time, or this is better to focus just in one component?
And normally it's a one compound. Isolating different compounds is the extremely complex thing. Okay, it's not possible in many ways. That is why the genetic transformation become relevant, because by which you can manipulate the expression of a particular gene that will induce the amount of this biomolecule that you are looking for, right? So that is why biotechnology become very relevant, because otherwise the cells are going to produce a lot of compounds at the same time, right? Some of them in a very high amount, some of them in a very low amount, but you cannot purify or isolate one out of hundreds, right? That's very complex. Even by analytical methods, a very laborious and very complex process. So that is why the transformation become relevant, and by which you can induce particular genes, or sometimes you can use the elicitation process. And this is for example, this is the elicitation. Elicitation means that it's a light, which I already showed you. It could be some kind of chemicals like jasmonic acid, for example. It can be used to induce the production of triterpenes. Okay, so if you know your compound is terpene or triterpene, you can use jasmonic acid. So you know that my compound is going to be in the higher concentration. Okay, so that helps you to at least divide the majority with the minority, or the minority with the majority, depending on the compound you are looking for. But yes, that is going to be produced in a very high amount. Okay, and obviously, there are the media optimization, by which you can also induce the production and inhibit the production of other kind of compounds. Precursor feeding, that is also important, very similar to the elicitation process. You can use some kinds of chemicals which could be a nutrient, which could be vitamins, that works as a precursor and induce, you know, the production, accelerate the production of a desired compound. Okay. And metabolic engineering. Okay, metabolic engineering is basically By synthetic biology, you can manipulate particular gene so that you can switch on and switch off the production of certain metabolites. And ploidy alteration is changing the chromosomes numbers, by which some of the productions can be inhibited and some production can be accelerated. So these are the method by which you can, you know, improve your method of production. Okay.
Can you repeat what was elicitation?
Elicitation is, as name suggests, elicit, like excitement, or elicit something by biological or non-biological factors. Okay. And what they does is they can induce the production of a particular metabolite or secondary metabolites in the plant. Okay. It could be light, it could be any kind of stress, it could be bacteria, it could be pH, it could be any chemical like jasmonic acid, anything. It could be anything, biological, non-biological, that can excite the plant, stress the plant in such a way that you can induce production of a specific metabolite.
Thank you.
Okay. Okay, question, doubts, guys? Yes, Juan Luis.
Yes, Professor, I have two questions. One, when you are making the suspension culture, the callus, the cells are going to be separated only with mechanical forces, or you can just also add like a detergent to...
No. It is possible, why not? But generally detergent is going to stress your cells, right? Depending on the method. There are so many variations in the methods, okay? So my answer will be, first answer will be no, and second answer will be yes, depending on the method. But I never use detergent in the liquid media, because detergent, as Marco already answered or question, no? That why they were using 2020 or 2000 or 2080 to release the alkaloids. It depending on the method how you establish, right? Every plant, every metabolites, and there are hundreds of variation that can be used and manipulated, right? So the answer is no and yes. Okay.
Okay, and my second question will be when you were like infecting the root with the rhizogene, how can you control the growth of the hairy roots? Because I think it's going to be pretty messy.
Well, you don't want to control normally. You don't want to control because you want them to grow in a very short period of time. Because, for example, I was doing a hairy root culture for 10 kg of roots. So I wanted to grow them very quickly, and it took like 6 months to reach up to 10 kilo of roots. So normally you want them to grow as fast as possible. Yes, once they because, you know, every living organism reaches up to a saturation point. Likewise, these roots also going to reach a saturation point where you can see the visible stress and visible symptoms, and you can understand that now cells need a fresh media because now they are not growing anymore. Okay, so everything will be depending on the condition you are going to provide, and definitely there will be always a saturation point. And if you want to maintain them for 1 year in a 10 kilo bioreactors, then obviously what you do, that you cut them and remove them. Also, that is also a normal practice. You just, you know, take out some of the cells and throw them, right? Because you don't want them to grow, okay? Because they are now hindering with your standardized process, okay? So depending on the protocol, how you can manipulate, but yes, in some cases, you want them to grow for a shorter period of time. Sometimes you want them to grow for a very long period of time, but yes, you can control them. And obviously, you can also find out some of the nutrients that can control the growth. You don't want them to grow so fast, right? So you can have less sugar, for example. You can add, manipulate some of the, sucrose is one of the best factor to control the growth, because it's directly control the growth. And you can also reduce the concentration of the vitamins, some amino acids, some proteins. Okay, so that also reduce the growth rate. Okay, there are many ways by which you can, you know, manipulate the growth. Okay.
I had one last question, teacher.
Yes.
The infection with the rhizogenes, it can only be on the roots or it can be on any part of the plant?
No, it can be on any part, but normally it is done with the shoots. So they have better impact and quickly. You can have in leaf, you can induce leaf, of course, without any problem. You can use shoots, you can use nodals, even cotyledons can be used. So basically any part can be used, but generally people prefer to use leaf or a normal plant which have little bit of roots, okay? But leaf is the, most of the time is the most suitable explant. This is the picture also shows you roots, leaf, sorry, right? Okay, just confirm. Okay, any other question, doubt?
Yes, teacher, I have one.
Yes.
So without the infection of the Agrobacterium rhizogenes, you cannot have the root hairs? Is not possible? Oh, okay.
No, you are going to have only normal roots, but hairy roots are not possible. Okay, so this is the exogenous molecule, right? Exogenous molecules, something which is coming out from the outside, obviously. And obviously this is just to show you the exogenous or endogenous, means it is being produced in the media, okay? So now here you can see the harvest the sample media, right? You are taking out the media, then you can identify of the molecules or the suspected other kind of molecules. Then you can find out what is the concentration and isolate and characterize your particular compound. And obviously the HPLC, HPTLC, chromatograph method, everything is become very related, okay? And then you upscaling and evaluation of biological. Here you can go with the research, you can find out the toxicity, you can do biological assays, in vitro models, in vivo models, animal testing, human trials, everything can be done here, or you can have the final products for the commercial purposes, right? And hairy root culture, so basically has a lot of application. Basically, we have discussed all of them, right? The recombinant protein production, right? Transgenic plants to induce production. Phytoremediation is another one by which you can create a lot of plants, right? With the new characteristics, and they can be used to absorb the toxins, right? From the soil. Molecular breeding, these hairy roots can be used to induce the breedings and accelerate the breeding, okay, like other methods. And obviously the phytochemical productions, and to understand the root physiology, right? Basic research. And to understand the biosynthetic pathways of the different kinds of compounds, because normally this is very important, and keep in mind, if you are using rhizogenous, sometimes they produce different compounds, okay? Sometimes they produce compounds that are not naturally produced, okay? That is why they call transformed root, okay? That is a normal phenomena, and possibly sometimes you find new molecules in the media, okay? That is also possible because bacteria has manipulated whole genome, and there are many surprises, okay? Question, doubts? All clear? I couldn't take the exam, so I will send you the Socrative by link, okay? So or maybe I will take on Monday, okay? I will take on Monday, okay? I will send you a grades, okay, by today and tomorrow. So you can check all the homework, and please check your feedback for the protocol designing, okay? Have you submitted? Everyone has submitted the protocol?
Not yet, teacher.
Not yet? Okay, please, tomorrow is the last day, okay? Please submit it because I will check by the weekend. Yes, Marco.
Teacher, I just have a question about the video we have to submit about our homemade cultures, because it is closed. We cannot submit